Hello everyone. In this lecture today, I'm going to talk to you about what is self-amplifying or self-replicating RNA. As the name suggests, self-replicating. Okay, self-replicating means that this RNA replicates on its own. So what is it actually? How actually does replicates? So first I'm going to talk to you about two kinds of RNA, that is self-replicating RNA or self-replicating RNA or self-amplifying RNA. Self-replicating RNA is also called self-amplifying RNA and messenger RNA. Okay, first I'm going to give you two examples. Okay, let's say that this here is the self-replicating RNA and this here is the messenger RNA. So what is the difference in between these two? In the self-replicating RNA, in addition to the coding sequence of our gene of interest, we also have the non-structural sequences of non-structural protein of the viruses, especially alpha viruses. Okay, for example, non-structural protein 1 to non-structural protein 4. So these are the sequences of our from the alpha viruses that form the replicase. Okay, this form the replicase or replication machinery. And so these replicase or replication machinery actually helps in the replication of our transgene. Okay, this is our transgene or gene of interest. Okay, so basically in messenger RNA we just have our coding sequence 5' and 3' UTRs. That's it. Here we don't have any these non-structural proteins from the viruses. Whereas in self-replicating RNA, we also put these non-structural proteins, non-structural protein sequences, sequences encoding for the non-structural proteins from alpha viruses to form the replicase or the replication machinery. And when inside the cell, this replicase or replication machinery replicates our transgene. Okay, this replicates our transgene. So here it is shown, for example, one example is here we have non-structural proteins sequences encoding for non-structural protein 1, 2, 3, 4, and this is the promoter sequence, and this is our transgene sequence. Okay, this is our gene of interest. This transgene sequence is our gene of interest. Okay, this is our gene of interest. Right, these sequences function as the replication machinery, and therefore they replicate this transgene when inside the cell. The detailed mechanism I'll explain later. So how these self-replicating RNA or self-amplifying RNA, how it replicates our gene of interest and produces more protein. So how it does, here is our self-replicating RNA. Okay, this is our self-replicating RNA. So this self-replicating RNA goes inside the cell, and in the cell, this self-replicating or self-amplifying RNA, it engages with the ribosome, because this ribosome is the one that does the protein production, right? So ribosome is the machinery in the cytoplasm of the cell that does the protein production from our RNA. So this self-amplifying RNA, it engages with the ribosome. So here the engagement happens. So then what happens? This self-replicating RNA is translated, because this self-replicating RNA has two things. In this part is for the replicase machinery, and this part is for our transgene or of our gene of interest. Okay, gene of interest. So the replicase machine, so what happens is that, okay, self-replicating RNA, it goes inside the cell, and in the cell it engages with the ribosome. And after the engagement with of the ribosome, because this self-replicating RNA or self-amplifying RNA, it has two parts, replicase machinery and also our sequences encoding for our gene of interest. So then our protein, target protein, will be translated. Okay, so from this part, okay, let's say this is this part, our target protein is translated, and from the remaining part, because we also have the sequences for replicase machinery, this replicase protein is translated. Okay, so first, this is very important, guys. Our self-amplifying RNA enters inside the cell, and in the cell it engages with the ribosome, and the ribosome translates from the sequence, from the self-replicating RNA, so our self-amplifying RNA, our target protein is translated because we have the sequences for that, and replicase machinery is also translated because we have the sequence for that. And what we need for the replication, as you know, we need these replication machinery for the replication. So when this replicase is translated, then what happens? This replicase machinery, it engages with our self-replicating RNA, because we have here again it engages with our self-replicating RNA. So replication machinery engages with our self-replicating RNA or self-amplifying RNA, and it then what happens? Then self-amplifying RNA is replicated. Okay, self-amplifying RNA is replicated as shown here. So when we have more self-amplifying RNA, and that will lead to the more production of our target protein. Okay, more production of our target protein. So I'll explain once again. So in self-replicating or self-amplifying RNA, so how they function. So self-amplifying RNA, they have the sequences for replication machinery and also for our gene of interest. They enter in the cell, and inside the cell, this self-amplifying RNA engages with the ribosome first, and from that our target protein is translated and replication machinery is translated. And this replicase machinery, what it does, it again engages with our self-amplifying RNA, and then what happens? The replication of self-amplifying RNA happens, and from that, when self-amplifying RNA is replicated, so from more self-amplifying RNA is produced, and from more self-amplifying RNA we get more protein. Okay, so more protein of our target sequence, right? So more protein is translated. So like I explained before, the self-amplifying RNA is a type of RNA that has many structural similarities to messenger RNA. It is linear single-stranded RNA molecule that is synthesized with 5' cap, 3' poly A tail, and 5' and 3' untranslated regions. So what is the difference between self-amplifying RNA and messenger RNA is that self-amplifying RNA are larger molecule. Like I explained before, they have both sequences for replicase machinery and also for our target gene, right? Target gene, and this here is the this is the replicase machinery. Okay, replicase machinery. So they are much larger sequences because they have the sequences that encode for extra proteins in addition to the gene of interest. And these 4 extra proteins, they are non-structural proteins, usually derived from these non-structural proteins. Then you may ask, okay, from where these which non-structural protein sequences, which non-structural proteins sequences encoding for which non-structural protein should be used, and those sequences are actually derived from alpha viruses that encode a replicase. The replicase enables the amplification of original strand of RNA molecule upon delivery into the cell. So this replicase functions when it's inside the cells, it gets translated, and then after translation it does the amplification of original strand of RNA, as I explained earlier. And this actually leads to higher amount of protein expression, and thus a minimum dose of RNA is required. Because what is the advantage of using self-amplifying RNA? So because we use one self-amplifying RNA and it is amplified, and we get a lot of protein. Right? This is RNA and this is protein. We get a lot of protein. So that means that we need low dose. This is a very, very big advantage. So we with the low dose of self-amplifying RNA, okay, low dose of self-amplifying RNA, we can achieve higher amount of protein expression. Right? So then the next thing is the self-amplifying constructs. They are used for gene delivery or vaccine applications, and they have, like I explained, they have been derived from alpha viruses, the sequences for replication machinery. For example, Venezuelan equine encephalitis virus, semlicky virus or Sindvis virus. These saRNA constructs contain 4 non-structural proteins, subgenomic promoter, and the gene of interest. So this gene of interest basically replaces the viral structural components. Okay. So by deleting the viral structural components, the RNA is incapable of producing an infectious virus. So basically, what is it actually? So if you say that this is a virus sequence and these are non-structural proteins and these are structural proteins of the virus. So then what is done when we make the construct for self-replicating RNA? We actually delete these structural proteins, and in place of structural proteins we replace it with our gene of interest. Okay, we replace it our gene of interest. So then we get this self-amplifying RNA construct like this. So here we have non-structural proteins required for replication, and here we have our gene of interest, sequences for the gene of interest. Okay, so this is just the repetition after delivery to the cytoplasm. Non-structural proteins from this replicase. This is generally RNA polymerase, and it replicates both genomic RNA and subgenomic RNA. That is our gene of interest. Each of the 4 non-structural proteins, they play important role in the formation of replication machinery, which is a complex and it is a multi-stage process. This replication is what leads to the higher expression than non-replicating mRNA. So the major advantage is with self-replicating RNA, with low dose, we produce higher amounts of protein, and we don't require higher dose of initial higher dose. We just, with a low dose, we can achieve a higher dose of protein. Okay, this is the major advantage of using self-replicating RNA over mRNA.